last speaker for this afternoon is Elliott Margulies from the National Human Genome Research Institute, Genome Informatics, Challenges in Comparative Genomics. Also, by the way, I should mention there is a wine reception after this talk. I don't, unfortunately, presume it's in the lobby. I don't know.
I think that's good.
Okay. Elliott.
How did you use this thing? This button? Oh, yeah, okay. Got it. All right. Well, it's a pleasure to be here. Thanks to the organizers for inviting me. We're going to take a 180 here. I'm coming from the biologist perspective. And what I hope to do today is more highlight many of the challenges that we face in biology and how I am hoping that statisticians can help us solve these problems. I'll also introduce you to some concepts that I think will be brought into more detail. I've had the great fortune of collaborating with Peter Bickel and his group, and he's, I know, going to go into a lot of detail from the statistical side on some of the things I'll be talking about. I also realize I'm up against two negatives here. You did not have a break before my talk, and you got wine coming to you right after I'm done, so we will try and move through this. Okay. I'm a genomicist, if that's a term you can call today. We have a complete human genome sequence, and one of the great challenges I think in the genomics community, a challenge that my lab tries to tackle is how can we interpret this genome sequence to try and identify functional elements. When we think about the human genome sequence, we know that roughly 1 to 2% of this genome codes for these things called genes, which code for proteins. They also do some other things. But we're very excited about trying to figure out what other functions exist in the human genome, where are they encoded in this genome, and how are they actually encoded? What are the sequences that encode certain elements? I thought for the purposes of this talk and this wide audience, it would be useful to classify at a very basic level what I mean by a functional element. And this is really, I think, in some ways, we can talk about enhancers, silencers, boundary elements, other types, our genes, protein coding genes, RNA genes, and so forth. But it all boils down to, I think, two fundamental concepts. And that's either a sequence of DNA that's acting as a binding site for a protein to come in and do something, and that could be one of many things, or it's a piece of DNA in the genome that codes for another functioning unit, namely RNA, and that RNA sequence does something, whether it translates to a protein or does some other regulatory sequence. Those are really the two fundamental components of what we're trying to search for when people like me come up here and broadly say, I'm trying to find functional sequences in the human genome. However, I think it's important to consider, and I know, so this non-coding variety is a particularly challenging component for a number of reasons. But it's important to recognize that, and I quote one of our former US folks over here in politics, there are these unknown unknowns, and we don't know what we don't know. And this is a real big challenge in the community. But I'm a proponent of a field called comparative genomics, and I think it will help us with these unknown unknowns. One of his better quotes, I think. So getting at this concept of functional sequences, I kind of separated them out in the previous slide into two components. This coding region component where we have a general understanding of the language. We know about exons and introns, this thing called the triplet code, which codes for these amino acids, and there are many of these complementary data sets like ESTs and cDNAs, very similar to what Lior Pachter showed you earlier today, that can kind of highlight where protein coding genes are falling, and indeed other RNA sequences. And this language of non-coding functional sequences is quite a challenge. It's in large part, we don't know what we're looking for. There are these short binding site motifs, algorithms such as the one that you and Bernie presented earlier this morning try to get at this signal-to-noise problem. And I think just kind of a pitch for these things called epigenetic chromatin modifications are starting to become a more promising way of trying to figure out where some of these non-coding functional sequences lie. So I'm going to talk to you a little bit about comparative sequence analysis, which I think is a very unbiased approach to trying to find functional sequences. So you come at it kind of from another perspective. Rather than saying, I'm looking for protein coding genes or I'm looking for enhancer sequences, you simply take a step back and say, I'm looking for anything that's functional. Once you find those, then you can now hand that off to an experimentalist and say, okay, what does this sequence now do? So I decided to take a step back here. Some other folks went through this earlier today, but how does comparative genomics really work? Well, first of all, everything living has DNA. This is, I think, sometimes quite fascinating when you try and think about this fact. And mutations in DNA are occurring randomly throughout the genome. But this is on a process that is in some ways very difficult to understand. It's not on the order of tens of years, hundreds of years, or thousands of years. We're really talking about millions of years. And this is really just a number, and it's a component to this process. If you think about genomes evolving, these random bits just acquire these mutations throughout millions of years. And the concept, and this was brought again earlier today, but sometimes I need to hear things multiple times before they start to sink in, is that a functional piece of DNA is less likely to tolerate a base change mutation because that change will likely abolish the function of that sequence. And this is just highlighted here. And this is why if we start comparing sequences across multiple species, the reason functional sequences will show up as being more similar is because there have been fewer mutations occurring in those particular sequences. So we can essentially develop various algorithms that can apply what we know about evolutionary theory to try and find sequences that have diverged less than we expect by chance alone. And it's for this reason that comparative sequence analysis can provide this unbiased view of where functional sequences exist. So of course this all depends on what we expect by chance with respect to finding functional sequences, and this all correlates with when we compare sequences, how distantly related the various species are. So for example, if I have a sequence of DNA that's 99% identical between two species, if the two species that I was comparing was between human and chimpanzee, well, that's actually what we might expect by chance alone. So that's not unusual. But that same 99% identical sequence observed between human and let's say rat would be very unusual, and therefore I'd be interested, and I would think that there's something interesting about that particular sequence. So these various algorithms that have been developed are called various things listed over here. I'm a particular fan of this one called SCAMS, which really isn't in the literature, standing for sequences conserved across multiple species, but the idea of publishing about SCAMS doesn't really bode well, especially when you're working in the US government. So this is just a quick list of some of the methods that have been now developed for looking for constrained sequences, things published, and so you may hear me fall into some lingo. In particular, I may talk about something called bin-cons, which is a binomial-based approach that we published several years ago for identifying conserved sequences across multiple species. So again, just to summarize this introduction here, when we use comparative genomics, we're essentially taking a highlighter, going across the genome, finding those bits of the sequences that are functional. In this case, we understand the language of the English language, so you can see the bits that we're trying to highlight. They say various interesting phrases if you're close enough to the screen. Okay, so my lab, and in general, I think the genomics field is interested in kind of two complementary questions. We're interested in knowing what are the functions of these various constrained sequences, but I think a more interesting question, particularly in light of the various genomic data that are becoming available, is to what extent are various functional sequences evolutionarily constrained. I think if we can start understanding, so to speak, from a true positive data set, what are the evolutionary signatures amongst various functional sequences, that may help us go back into the genome and help computationally characterize other constrained sequences that we've identified. So I'd like to introduce you to the ENCODE project, which is a project that I was fortunate enough to be working on, in particular in the comparative sequence analysis efforts, and you and Bernie kind of led many of these analyses that led to this publication in Nature, and also a series of papers in this issue of Genome Research. So the ENCODE pilot project started with 1% of the human genome and brought together both computational folks and experimentalists to hammer at this 30 megabases of the genome with redundant technologies and try and figure out where certain functions were encoded. And it gave us a very deep data set to do some unique things that we couldn't do genome-wide. I thought it would be helpful to, if this will come up, to let you know essentially which 1% was picked for studying. You could imagine lots of folks want their particular gene of interest. Well, essentially half of the 1% was picked because of some sort of prior interest or already deeper data sets available. For example, the alpha globin and beta globin genes, these are regions of the genome that have been tested from an experimental perspective very deeply. But the other half of this 1% was selected randomly based on two stratifications. At the time that this project was started, it's kind of hard to believe, but there was only human data and mouse data with respect to vertebrate comparative genomics. And so we stratified the genome into low, medium, and high non-coding conservation between human and mouse. And then we also looked at gene density, again, low, medium, and high. So you essentially have these 9 strata, and random 500 kb regions of the genome were picked. The only other constraint on this pick was that they were picked in regions that were essentially finished in the human genome at the time, because at the time that we did this analysis, it wasn't complete, or essentially complete, as the term has been brought up. So with respect to the ENCODE, this is a little bit frustrating with animation, I apologize. There were a whole host of various data sets being generated from gene annotation work, comparative sequence analysis, which I'll get into a little bit, the promoter identification, looking at DNA-protein interactions. This is something I think you'll hear from others in the talk this week. In particular, Greg Crawford's going to talk about some work that he's done, and I'll highlight some of the data that actually his group had generated, and also looking at RNA expression. And this gave us kind of an unprecedented view to really cross-compare these various data sets. And in particular, we were interested in identifying these evolutionarily constrained sequences and then figuring out what proportion of these sequences overlap many of these annotations that were identified by the ENCODE consortium. And what we essentially found was a 5% of this 1% of the genome. We had a really high confidence, and I won't go into the details, that this 5% was under strong purifying selection across all mammals. And when you look at the breakdown, roughly a third of those constrained sequences overlapped known coding regions. And then what we were able to do within the ENCODE consortium, which we were never able to do before, was look at other non-coding functional sequences and see to what extent these evolutionarily constrained sequences overlap this data set. Because there was never a coordinated effort to deposit many of these non-coding annotations into some sort of universal database, nor was there evolutionary deep data to identify with high confidence constrained sequences. So this 1% was a really neat data set to analyze. And we identified roughly another 20% of these constrained sequences as overlapping experimental annotations. Now, there was still 40% that was unaccounted for. Again, I'm not too concerned about this. It goes back to Donald Rumsfeld. There are things we don't know, and I'm sure that 40% comprises that. But something that was actually quite interesting was the fact that there was and I'll show you this in the next couple of slides before I go on a little statistics detour, although this is coming from a biologist, so bear with me, was the fact that there were a number of experimental annotations that showed no evidence for being under any evolutionary constraint the way we detected this. And we found this particular mechanism of looking for overlaps between two different data sets kind of a challenge. And from a non-statistics perspective, what many biologists tend to do is take their data set and randomize it 1,000 times and see how well the overlap looked amongst this 1,000 randomizations, and then how well the real overlap looked with the randomized overlap. And this worked to some extent, but we really wanted to come up with a more formalized way of doing this that could take into account various complexities in the data set and in what we know about the genome. So taking a step back, we looked at this overlap in kind of two different ways. One was what I refer to at the base pair level, and another was at the region level. And that's what's highlighted here. On this screen, it's yellow and blue. Over here, I think it's 2 shades of green. this orange data set over here represents some hypothetical set of constrained sequences. This represents some sort of hypothetical experimental annotation. If you look at the base pair level, you're asking how many bases of this entire experimental annotation overlaps these constrained sequences. And overall, while some annotations show more or less overlap and some show none whatsoever, overall, in this hypothetical example, 25% of the bases are shown to be constrained. But we can also do kind of a binary yes-no. And this we found to be quite revealing in that for this hypothetical example, you can see that 3/4 or 75% of these experimental annotations, whoops, sorry about that, show up as being evolutionary constrained. And so how did we determine the overlap here? Well, in collaboration with Peter Bickel's group, we developed this method called the genome structure correlation, or GSC. And essentially, this gets at in a more formal way, given two types of genomic features, how significant is the positional overlap between them? And what Peter Bickel's group developed was essentially treating the genome like two bivariate time series, where time is kind of equivalent to your position in the genome. It has a number of underlying assumptions which we can generally hold true for the genome, and then essentially develop this block bootstrap method to look for significant overlaps and come up with more formal p-values. And I tried to highlight this in graphic form in this next slide. So let's say you have these two features on the genome, feature 1 shown on the top of the black line, feature 2 on the bottom. You're randomly pulling sub-blocks from this genome and then actually reversing, you're swapping kind of feature 2 over here to create what we internally call this kind of dummy overlap. And then that gives you, by doing this a number of times and then normalizing this distribution, you can calculate some statistical measures that give you some level of confidence as to whether or not your actual overlap is biologically significant, or I should say statistically significant. Then we have to go and figure out if it's biologically significant. And this was quite helpful. It takes into account the fact that at a large scale, there are a certain genomic feature might be clustered in one area and sparse in another area. And it takes that also into account the fact that both feature sets might be that way, because many times we find that certain, as Ewan alluded to this morning, many things tend to correlate close to genes because for various effects, and we had gene dense and gene poor regions, and so this type of a method was quite helpful in figuring this out. And stay tuned for the statistician's view on this statistical method in the coming days. So this is now showing you the overlap of constrained sequences with various encode annotations. You can kind of ignore the specifics down here. Essentially, we have 3 sets of data, things involved in RNA transcription, things looking at regions of open chromatin, which we think are important for various functions, and things looking at DNA-protein interactions. The top part over here is at the base pair level. The bottom part over here is at the region level. Here are your error bars calculated from this nifty new method that we were able to use. And something that biologists historically look at, because again, it's something that we know what to expect, are these coding regions, where we see the vast majority of these coding regions overlapping constrained sequences, both at the base pair level and at the region level. And I think we naively assumed that going into various non-coding functional sequences, we would see the same type of overlap, but it was indeed not what we saw. And in fact, it dropped to being roughly around 50% of the features seem to have this type of an overlap. And many times it was significant at kind of the feature level, but it was barely significant or not significant at all at the base pair level. And this kind of led to a number of interesting questions and hypotheses that were brought up as to why was there not this lack of correlation? Why did we actually see many experimental annotations with no evidence whatsoever of being under evolutionary constraint? One hypothesis which we showed to be true, was that sometimes the annotation was actually larger than the functioning unit. This is due to the resolution of the microarrays that were used at the time for generating many of the data. Another important concept is that we were identifying constrained sequences that were constrained throughout all mammals. It's certainly possible that many of the functions that were identified were constrained in only subpopulations of species, and this is something that we need to take into account. And it's also possible that there is evolutionary constraint that isn't being detected with direct evidence, with direct understanding of the primary sequence alone. Another way of saying this is that we don't completely understand the parameters of evolution on DNA sequences and what constrains certain bases versus others. And I'd like to actually talk about this aspect a little bit for the second half of my talk and introduce you to some work that my lab is doing to try and enhance sequence constraint algorithms and take into account more information than what has typically been done in the past. So this is an example of a multiple sequence alignment for a region that we consider under evolutionary constraint. What you're looking at is, this is the human sequence, the sequence of the human genome in a particular region, and these are a list of a number of other species down along this so-called y-axis, and every time you see a dot, that means that the sequence was found to be identical between human and the other species. Anytime you see another base over here, that means that that base has changed. And just by eye, you can kind of go along this region and see, well, yeah, this particular region is under purifying selection. There are a lack of substitutions in this particular region. But what if there was something else about the DNA that allowed certain substitutions to occur, but yet the function might still be maintained? This particular method might not necessarily pick that up. So I want to introduce you to a concept that we've been working on in the lab. This has been in strong collaboration with a DNA chemist, Tom Tullius, who's at Boston University. And we're hypothesizing that there might be certain substitutions that are more tolerated for preserving function and than other substitutions, and meaning that certain base changes might not affect the DNA structure as much as other base changes. And we know that DNA structure, in particular kind of non-base specific contexts, can be very important for DNA-protein interactions, which is kind of one of the two main categories of functional sequences that I proposed in the beginning of this talk. And so the question is, how can we measure DNA structure? Many times the bioinformaticists will sit down at a computer and look at DNA sequence as a string of letters, and we frequently do that. And you forget that DNA is a three-dimensional molecule that has structure to it. And I wanted to come up with a way that maybe we can incorporate this information into our sequence constraint algorithm. So the question is really, can this structure information be used to improve comparative sequence analysis? So there are a number of different methods that can be used to identify the structure of DNA. DNA crystallography is kind of or crystallography in general is kind of the de facto standard, but this is very expensive, labor intensive, and not quite scalable to a kind of a genome-wide level. There are complete computational methods that work and that are purely based on simulation. DNase I sensitivity is another method for looking at DNA structure, but it's not a single base resolution, nor is it at the measure of molecular bonds between protein and DNA. This is something that I'll leave Greg Crawford to talk about, who's the expert on this. And what I would like to introduce you to is a chemical reaction called the hydroxyl radical cleavage, which is based on DNA in solution, and I'll show you that it actually correlates with crystallography measures, which is kind of the de facto standard, and we have a relatively quick and accurate prediction algorithm that can be used. So DNA footprinting and this hydroxyl radical chemical reaction was developed by Tom Tullius, and if you read this paragraph, it's quite interesting how much the high-level questions in genomics were known even many years ago, and we're all kind of touting the same questions even today. So he went on to show that the intensity of this hydroxyl radical cleavage was really governed by the 4 and 5 prime hydrogens, which are highlighted on this kind of molecular structure crystallography structure of DNA, and that the more accessible these 4 and 5' hydrogens are, the more intense the breaking of the bonds are going to be there, and that this is not correlated with the actual base composition. So it's independent of the base pairing that's going on there. So just to kind of zoom in, what he's measuring with a hydroxyl radical reaction here is kind of the solvent accessible surface area of DNA. So if you can just imagine zooming into this particular base pair showing the 4 and 5' hydrogens, these are more accessible just because you can see more of the red and the blue, but this is also quantitatively measured using computer programs on the DNA structure. This is a particular hydrogen that's less accessible. And so these are the types of things that we're trying to now measure and bring onto the genomic DNA sequence. So they've actually developed a database and have taken pieces of DNA, undergone this chemical cleavage reaction, and measure essentially the intensity at a particular base position, where bases like this are more accessible, bases like this are less accessible. Okay, and so what this slide is showing you is that these patterns can all be correlated back to the actual structure of DNA. So this, let's see, so a couple of these colors aren't showing up. So this particular graph over here, this particular line, is showing you the experimental data with respect to the hydroxyl radical intensity. So this particular region, this is it's called the Dickerson dodecamer. It's one of the famous 12 base pair sequences that was first crystallized and has been extensively crystallized and studied. And this is what the cleavage intensity pattern looks like. When you actually computationally examine the crystal structure of that dodecamer, the pattern of solvent accessibility of those 4 and 5' hydroxyls looks like this, which is very similar to the pattern that you see with the hydroxyl radical cleavage pattern. And then the other exciting thing is that with this computational algorithm that looks into a database of sequences that have been analyzed with this experimental hydroxyl radical pattern, you can see that this pattern also pretty much also follows what is known about both the crystal structure and the experimental data that was used to generate from the prediction algorithm. So we thought that using this information would be quite helpful, particularly because what I'm showing you here are the cleavage intensity patterns for 2 different sequences. It's 10 nucleotide sequences that are 0% identical. So you can see that you can have 2 sequences that have 0% similarity, yet they can have similar DNA structure. The idea being that maybe for certain protein DNA interactions, it's not the actual base pairing that's important, but it's the overall topology, structure, and solvent accessibility of various components of the DNA structure that are important. Now, granted, this is an extreme example found in a database. I'm going to show you some more real examples in a moment. So another concept that I think is important into dealing with this type of an algorithm is that certain substitutions appear to be more tolerable than others. I'll throw out this term tolerable. And this is an example here. We've taken an 11-mer sequence and then changed the middle base pair to be any one of A, C, G, or T. And what you can see is that for the most part, the structure pattern seems to follow as being quite similar. Whereas you can take another DNA sequence, change the base right in the middle, and each base position has a much more dramatic effect on the structure pattern over here. And so what if we can capture this information and know what substitutions result in something like this versus what substitutions result in something like that? Maybe we can improve some of these sequence constraint algorithms. And of course, the statistics behind trying to do this are also a challenge. So a final kind of aspect to why we think this might actually be revealing something important is shown here. So what Steve Parker, who's the graduate student in Tom's lab who's been working on this project, has done is he's gone into the literature, looked for experiments where people have done saturation mutagenesis on various DNA protein binding sites and looked at their correlation with changes in DNA structure. And so for this particular paper, they did saturation mutagenesis on this particular DNA sequence where they changed all of the different bases and looked at the protein binding affinity using gel mobility shift assays to see how much better or worse the particular mutated binding site did. And Steve focused on the minor groove area, which is known to have non-base specific important interactions between this protein and DNA molecule, and essentially summed up kind of the Euclidean distance measured structure pattern for various mutations, and then also what he's calling KDREL, which is the relative DNA binding. So the increased number means the fold difference between the wild type and the mutated version over here. And a couple of examples are shown by mutating a couple of positions in this minor groove where you can see strong perturbations in the DNA structure pattern, whereas others seem to have very similar DNA structure patterns. And if you plot a lot of these data, what you can see is a modest correlation between the DNA binding affinity and the Euclidean distance. So in other words, the farther away you are from the wild type version of the DNA structure and the worse you are binding compared to the wild type, there tends to be a trend over here. So now with all of this information, we decided to build this into a sequence constraint algorithm and develop what I would call a structure-informed sequence conservation score. And what this is, what I'm hoping this will show you that we can do is now with some very statistical confidence, we can try and improve our ability at detecting more weakly constrained sequences that we might not have been able to detect just knowing, just treating all substitutions equal. And in an effort to compete with the Europeans on the naming scheme, we have called this algorithm Chai. He likes chai lattes, so he came up with this while drinking a chai latte, and that's how he got it. So the steps in this particular constraint algorithm is to take a multiple sequence alignment and convert it into these structure scores, which relate to the DNA solvent accessibility. And then we create a similarity score, which is based on the Euclidean distance between these measures. So we go into a window and are asking essentially how similar are these row of numbers with these row of numbers over here. And we weight all of these species based on their distance from the human sequence. And then we can come up with a score. Now, I didn't realize that the speaker before me was going to really go into detailed analysis of the false discovery rate approach. But I'd like to bring this approach to you in how we use this for analyzing multiple alignments, which is a real challenge in biology. Coming up with what we think is neutral or shouldn't be detected is always a challenge when you don't know the complete story. And as was also brought up earlier today, knowing what should be detected is in totality is also a big challenge. So what we do for identifying and trying to kind of level the playing field with a number of different algorithms is come up with a data set that we think represents neutral evolution. And because this data set is, we're generating it before any algorithm approaches trying to do any sort of analysis, it sets a common ground for being able to compare different algorithms to each other. So we start off with a kind of a real multiple alignment, and we mask out regions that are kind of low phylogenetic diversity. And then we kind of go into the rest of that alignment and kind of bootstrap out various alignment columns and create this kind of null or neutral alignment. Now, it's certainly not perfect, and you can argue going after this in different approaches, but again, it gives us at least a relative metric for comparing various algorithms to each other. And so what we attempt to do is use some sort of algorithm, whether it's this binomial-based approach or I should have changed this to Chai, and then figure out what fraction of the regions are being identified with these particular algorithms, and then you can also plot that against the false discovery rate, which is how much sequence is being, essentially how much sequence is being detected in this null or neutral alignment that we created. And the first thing you'll notice is that kind of depending upon, no matter what false discovery rate we use, this new structure-based, structure-informed algorithm is detecting roughly twice as much sequence being under evolutionary constraint as some other standard methods are. And we kind of focus in on that 5% level. And so what is this now 12% that's being identified? Well, it turns out that roughly half of this is only detected by this new structure-informed algorithm. Roughly half of it is detected by both algorithms, and then you have this little bit that seems to be sequence only. Whether or not those are false positives or truly these rare situations where very similar sequences can have very different structures, still remains to be determined. This is an example of now the kinds of elements that we're identifying in multiple sequence alignments. Here's another example of one of these primary constrained sequences with very few substitutions. This is a region that has many more substitutions in it that seems to be identified as being under constraint when we add in this structural information. When we look at the proportion of the constrained sequences that overlap various functional annotations, looking at this binomial-based algorithm, you can see that roughly a quarter to a third of this represents a known coding sequence, very similar to, this is a slightly different data set, so it's differing from some of the graphs I showed you earlier. And then 13% overlap some non-coding functional annotations with a little more than half being unaccounted for. What happens when you increase the amount of sequence? You're essentially reducing the proportion that's overlapping coding sequence. Everything else kind of stays the same, and also increasing the amount of non-coding sequence. This fraction is really being reduced not because we're detecting fewer of these coding regions, but because we're detecting more non-coding regions. So this is now a graph showing various non-coding and coding annotations with using 3 different methods now. We use this binomial-based conservation program, which has been kind of the hallmark and what's been used in the past. We also use a binomial-based method where we've relaxed the false discovery rate such that the overall coverage is roughly the same as it is with using this Chi method that's done at a 5% false discovery rate. And what you'll see for coding sequence, oh, and then of course we have our great statistical algorithm that shows what we expect by chance, the null distribution shown over here by these error bars. And so for coding regions, we're not doing that much better, and this is something that we might expect because we've already done quite well, and we also know that that the primary sequence is quite important in coding for these coding regions. But these things like DNase I hypersensitive sites, these things called fair sites, which I'm sure Greg is going to go into detail about, we can actually detect more sequence as being evolutionary constrained. And granted, you're taking a big jump when you go from 6% to 12%, but even when you're comparing something that only takes into account this primary sequence versus something that is structure informed, you're also doing a little bit better at this. These things, so complementary to kind of a null data set are things called ancestral repeats, which are we think are largely neutrally evolving. And what's interesting to see is that there's kind of a paucity of these elements in ancestral repeats. And just to show you that we also generate kind of a random data set, and it falls right within kind of expectations over here. Another thing that we're able to do is just pull out what we call the structure only. So the elements that were only detected with this new algorithm called Chi. And what you'll see is that for coding regions, we actually see much less than we expect by chance. And we think, again, this is because for protein coding regions, we know that the structural component is not as important. But even when we take these structure-only regions, we detect a very large number of these hypersensitive sites and other regions that are believed to be important for in non-coding functional annotations. And again, the random or null distributions kind of check out as well. So just to give you an idea from kind of a DNA logo perspective, if you look at a DNA logo of a region that's under primary sequence constraint, it kind of looks like this. You have very strong signal, lots of information in every base. if you look at a piece of DNA that we think is under neutral evolution, the bits of DNA have kind of less information in them. And I think some of these regions that are detected with the structure-informed algorithm kind of fall in between these two methods, but there's still some level of constraint here. But I think the key is that we're now able to add more information to the algorithm, so at the same level of statistical confidence, we can now detect more sequence under constraint. Just a final word here that we think we've hit on something that is, in fact, biologically relevant. We went into a database called FenCode, which is, tries to map a bunch of mutations known in the genome that are thought to cause disease. And this is a particular mutation that occurs upstream of the hemoglobin gene, where if you look at the structure pattern of the wild type sequence versus the mutated sequence, just this one base change causes a dramatic change in structure over here. And this is also a mutation that's known to cause disease. We've tried to go in and look at a number of SNPs, try and separate SNPs, neutrally evolving SNPs versus SNPs that might cause disease, and we clearly do see a shift. The more dramatic structure changes are occurring in those mutations that seem to cause disease. But this is actually a real big challenge in databases because it's not very easy to find a database that's completely just filled with disease-causing non-coding mutations. Just to say, we're also going into the lab and taking some of these CHI elements and looking at ones that overlap other data sets that seem to suggest there's enhancer sequences. This comes from a paper from Bing Ren where he's done some epigenetic chromatin modification and found regions of the genome that seem to predict enhancers. We then went in and found these regions and found ones that overlapped only these CHI elements and found that some of them do in fact show enhancer activity in our hands. And where we're planning on going with this is to now take these particular sequences, mutate them such that they don't change the structure very much, and see if we can maintain this enhancer activity versus mutating them where they do change the structure dramatically. And I think that'll bolster the evidence that the structure is indeed important here. So just to wrap up, I think I've shown you that changes in the DNA structure can have various functional consequences. We've developed what we're now calling kind of the structure-informed sequence constraint algorithm, which seems to detect twice as much constrained sequence at the same statistical confidence. And it also seems to correlate better with many of these ENCODE functional annotations. And we're currently undergoing many of the various experimental validations with these data sets. And so just to acknowledge the ENCODE project for providing these data, I work closely with the NIH Intramural Sequencing Center generating lots of comparative sequence data. In my lab, various folks contributed to this analysis, and at BU in particular, a lot of the structure work was all Steve Parker's work, who's a fabulous graduate student. So I hope I've given you an appreciation before you get to have some wine about the statistical complexities in both trying to analyze DNA sequences under evolutionary constraint and kind of a teaser into Peter Bickel's talk about taking two genomic feature sets and trying to figure out if they're statistically significant with respect to their overlap. So with that, I'll take any questions, and thanks for your time. Ewan.
So you know the question that's going to come, because the structural model that you have isn't direct structure, right? It comes from this database. It's like a 12th order Markov complicated recipe.
Right.
Yeah. Mm-hmm. It's all computational at the end of the day, and in theory, this is So have you rotated the numbers in that underlying database, and does it go away? That's for me always the
right.
I mean, in some sense, is this just the world's most complicated way of measuring GC content on the planet?
Right.
And which it could be, right?
So it doesn't go away, and the reason why, but it gets reduced.
Okay. It doesn't go away, right?
Exactly, so you've actually been privy to earlier versions of this research. That I don't know how many others have heard me talk about this before, but we have kind of had a paradigm shift in what we think this is doing. in that I don't think this is picking up DNA sequences that are not under primary sequence constraint. I think this is improving an algorithm identifying those substitutions that could be tolerated with respect to structure versus not tolerated. The challenge with doing this experiment that you've proposed about swapping everything is that the primary sequence is in fact important in this underlying algorithm, and you can't tease out. You have to look for the kind of the most extreme cases where there are very weakly constrained sequences where the structure is the thing that's really contributing to this analysis. And I kind of attribute it to the fact that we have this amino acid triplet codon table. If you swapped all of the identities within that triplet codon table and then went back to a genome and said, go find me things that are similar, it's going to pick up the same things that are similar, but it's going to be gibberish, right? Because the wrong amino acids are going to be linked up to the wrong triplet bases that are similar. And that's in essence what we're doing, what we would be doing if we did that randomization that you're suggesting.
So I appreciate. I mean, the fact that it's slightly lower, though, it tells you that the way the numbers are laid out is, at least, you know,
giving you some information.
It gives you some information, but I mean, it's just one of those confounding things where in some sense you have a very complicated nth order Markov model that sort of summarizes itself into some numbers, which you then use onwards, and there's a viewpoint where you know, so I agree that this is sort of picking up these. I agree with your analysis, your kind of instinct. It'd be very nice to see those results. Though, do you have them?
I may have anticipated this.
I always ask this question.
Yeah. Right. So we took this tetramer lookup table.
Just rotate the numbers.
Yeah. Randomize them here.
Okay. Brilliant.
Yeah. You're making me look good here. Okay. And so okay. So when you look at this is kind of still work in progress, but the overlap between kind of real elements and these randomized elements between that have been under sequence constraint and structural constraint, you kind of get this 96% overlap. When you look at kind of the structure-only regions, the number drops to kind of 93. And then when you take what we call extreme structure, which is you take out kind of the 12%, the reduced false discovery or the increased false discovery rate, primary sequence-based elements, so the same same coverage level. So stuff that's only picked up by this structure-informed algorithm and not picked up by any sequence constraint algorithm, no matter how far you kind of reduce or increase the coverage, the number drops even further. So as
wait a second, 90% of those things get picked up when you randomize the numbers.
That's right.
So this implies that effectively, if you do any analysis based on tetramers, you should get this as well. Somehow we're just, tetramer analysis is more powerful than single base pair analysis would be the interpretation of this slide. Give or take the 10%. I mean, I'm impressed that there's a 10% shift, but in some sense, this is saying that tetramer analysis is more powerful than...
Right. But you're right, but what we're also doing now is now linking up that tetramer analysis and showing, I think, some interesting biological tidbits that correlate with known mutations, known DNA protein binding, and what we should do is reshuffle things up and see how those correlate. Yeah. Yeah.
How does this, if at all, relate to RNA structural concentration?
Well, some of the data sets that we've looked at are non-coding RNA elements, and in large part we haven't seen a significant overlap with them, but we need to go back. I mean, there's been kind of a plateau in the ENCODE group, but I know that this area has really been looked at, and I'd love to get my hands on a new data set that would analyze that. Yeah.
Can you somehow subtract the nucleosome packing pattern or something like that? I mean, some background structural conservation that is sort of spread out over the entire genome. I mean, there's a big literature on how promoters bend and all that.
Yeah, so this is some...
You talk about something specific in most of your talk in terms of function. Mm-hmm. And the nucleosome positioning code, of course, it is organized, and correlated with the function patterns and the promoters, but it is also everywhere.
So I think what you're getting at is something that's still in question with respect to whether there's some sort of nucleosome ordering that's encoded within DNA. I know that there's been some papers out there that have said yes. Other papers have kind of refuted this. But what we're looking at is something that's inherent to the naked DNA structure. So it's once you've actually unraveled the DNA out of its nucleosome structure, and you have these protein DNA interactions occurring to cause some sort of functional phenomenon, that's the kind of thing that we're measuring. And the reason why we think this is important is because it's been shown that most protein DNA interactions that are occurring in the genome are actually occurring on naked DNA. It's happening once you've unwound it. So once you've gotten these regions of open chromatin, that's when these things are happening. These protein DNA interactions are not necessarily occurring while the DNA is all wrapped up.
No, but you're sort of measuring the grand total. I was wondering whether you could subtract what stemmed from, for example, nucleosome positioning and then see what is left.
Not sure I follow, but
Let's have it over wine.
Yeah, let's do that. All right. Thank you.
Okay.